In this video, we'll talk about glyoxylate cycle. The name glyoxylate is derived from the intermediate compound that is formed during this reaction. So, an intermediate compound, that is glyoxylate, is formed in the glyoxylate cycle or in the process of a glyoxylate cycle. That's why this cycle is known as a glyoxylate cycle. This glyoxylate cycle is a modified or variation of Krebs cycle or citric acid cycle or TCA cycle or tricarboxylic acid cycle. So, what is the meaning of variation of tricarboxylic acid cycle? Some of the reactions of the glyoxylate cycle is same as that of the Krebs cycle or citric acid cycle. That's why this glyoxylate cycle is a modified or variation of the Krebs cycle. This glyoxylate cycle aims, or the purpose of glyoxylate cycle is to produce succinate from the acetyl CoA. So acetyl CoA, which is formed mainly because of beta oxidation of the fatty acid, will convert into succinate in the glyoxylate cycle. Later, this succinate will produce carbohydrate upon undergoing gluconeogenesis, which is a main purpose or aim of the glyoxylate cycle. So, in glyoxylate cycle, acetyl CoA is converted into succinate. Later, this succinate undergo gluconeogenesis to produce carbohydrate. We'll see how succinate is formed from the acetyl CoA while discussing the reaction of a glyoxylate cycle. Before that, we'll talk about where this glyoxylate cycle takes place. This glyoxylate cycle is seen mainly in plant, fungi, protist, bacteria, etc. And these reactions occur specifically in the glyoxysome. As you all know, glyoxysomes are the modified peroxisome. So this glyoxylate cycle is present or it occurs in the glyoxysomes of plant, fungi, protist and bacteria. So these reactions takes place in the glyoxysomes of these organisms. And the glyoxylate cycle is totally absent in the animal except for the nematode. In nematode, only during the embryogenesis these glyoxylate cycles is seen. So only during the embryogenesis of the nematode glyoxylate cycle is seen. Apart from that, none of the animal shows this glyoxylate cycle, and the main reason for that is absence of the key enzyme of glyoxylate cycle, that is isocitrate lyase and the malate synthase. So these two are the key enzymes of this glyoxylate cycle. That's why glyoxylate cycle is not present or totally absent in all animals. So that is about where this glyoxylate cycle is present. Now we'll talk about the significance or importance of this glyoxylate cycle. This glyoxylate cycle play a very important role in the plant, mainly during the germination. As you all know, during germination, the lipid that are stored in the seeds will be converted into carbohydrate, because the seedlings or the germinating seed will not photosynthesize to produce carbohydrate. That's why the seedlings completely depends on the lipid that are stored in the seeds, and these lipids will be converted into carbohydrate mainly through this glyoxylate cycle. Later, this carbohydrate will be utilized by the seedlings as a carbon source or source of energy. So conversion of lipid into carbohydrate during germination is done mainly by glyoxylate cycle. That's why glyoxylate cycle play a very important role in the plant. So that is about the plant. If we talk about the microorganisms like protist, bacteria and all, these organisms can grow in a medium containing simple carbon sources using this glyoxylate cycle. Even in the absence of complex carbon sources like glucose or any other carbohydrate, these microorganisms can grow or can survive using this glyoxylate cycle. So they will utilize as a simple carbon sources using glyoxylate cycle in that medium, and they will survive or they will grow. So that's why glyoxylate cycle play a very important role in plant as well as in the microorganisms like bacteria, protists and all. So that is about the significance of this glyoxylate cycle. Now we'll talk about the actual glyoxylate cycle, or what are the reaction that takes place in the glyoxylate cycle. As I already told, this glyoxylate cycle is a modified or variation of the Krebs cycle, which means some of the reactions of the Krebs cycle is present even in the glyoxylate cycle, or some of the reactions of the glyoxylate cycle is same as that of the Krebs cycle. For example, the first step where the acetyl CoA combines with the oxaloacetate to produce citrate, and this reaction is same as that of the Krebs cycle. In the glyoxylate cycle, this acetyl-CoA is formed as a result of beta oxidation of the fatty acid, and beta oxidation of the fatty acid takes place in the glyoxysome itself. So, as a result of beta oxidation of the fatty acid, acetyl-CoA is formed, and this acetyl-CoA will not oxidize in the Krebs cycle, or it won't enter Krebs cycle. Instead, it enters only glyoxylate cycle. So, acetyl-CoA, which are formed as a result of beta oxidation of the fatty acid, will enter only glyoxylate cycle. So that is about the acetyl-CoA. If we talk about oxaloacetate, this oxaloacetate is actually from the mitochondria. So from the mitochondria, oxaloacetate enter glyoxysome. So during the entry of oxaloacetate from mitochondria to glyoxysome, it will be converted into aspartate, because oxaloacetate cannot cross mitochondrial membrane. That's why it converted into aspartate. So first, the oxaloacetate in mitochondria is converted into aspartate, and this aspartate crosses the mitochondrial membrane and it enters the glyoxysome. Later in glyoxysome, this aspartate is converted back into oxaloacetate. So now this oxaloacetate in the glyoxysome combines with the acetyl-CoA, which are formed as a result of beta oxidation of the fatty acid, to form a citrate, and this reaction is catalyzed by the enzyme known as citrate synthase. So oxaloacetate combined with the acetyl-CoA in the presence of enzyme known as citrate synthase to produce citrate. In the next step, this citrate is converted into isocitrate in the presence of enzyme known as aconitase. Again, this reaction is same as that of the Krebs cycle. So in the second step, citrate is converted into isocitrate, which is catalyzed by the enzyme known as aconitase. In the next step, in the citric acid cycle, this isocitrate is converted into alpha ketoglutarate, and alpha ketoglutarate is converted into succinyl-CoA. So these two are the decarboxylation step. Decarboxylation means carbon dioxide is released in both the step. But in the glyoxylate cycle, these two steps are bypassed, or decarboxylation step is skipped in the glyoxylate cycle. Instead, isocitrate is directly converted into succinate. So, in glyoxylate cycle, these two decarboxylation steps are bypassed to save two carbon atom, and that is saved in the form of a glyoxylate. So in the next step of glyoxylate cycle, isocitrate is splits or lyase to form succinate and glyoxylate in the presence of enzyme known as isocitrate lyase. So isocitrate is converted or split into two compound, that is glyoxylate and succinate, in the presence of enzyme known as isocitrate lyase. So because of this glyoxylate, this cycle is called as glyoxylate cycle. So in this, loss of two carbon in the form of decarboxylation is skipped or bypassed, and two carbon is saved in the form of glyoxylate. So that's why it is known as a glyoxylate cycle. In the next step, this glyoxylate is converted into malate in the presence of enzyme known as malate synthase. So in this case, isocitrate lyase as well as malate synthase are known as key enzyme of glyoxylate cycle. So glyoxylate is converted into malate in the presence of enzyme known as malate synthase. In the next step, malate is converted back into oxaloacetate in the presence of enzyme known as malate dehydrogenase, or conversion of malate to oxaloacetate is catalyzed by the enzyme known as malate dehydrogenase. So very important part of the glyoxylate cycle is it skipped decarboxylation step to save two carbon, and that carbon is saved in the form of glyoxylate. So in one term of the glyoxylate cycle, 2-acetyl-CoA is utilized to produce one molecule of succinate, and this succinate is of great significance because in most of the cases these succinate molecule undergo gluconeogenesis to produce glucose or the carbohydrate, whereas in some cases this succinate may even enter the Krebs cycle or a citric acid cycle, or it may be used as a precursor to synthesize some of the amino acid. So in one turn of the glyoxylate cycle, mainly 2-acetyl-CoA is utilized to produce one molecule of succinate, and the glyoxylate is used as an intermediate product. That's why it is called as glyoxylate cycle. So that is about the glyoxylate cycle. I hope this video is useful. If you like this video, please hit the like button and subscribe to my channel. Thank you.